let's welcome Dr. Sanchez, and we have to listen to this topic about the autophagy, which will be the molecular mechanism on neurodegenerative disorders. Okay, so I'll pass the time to Dalinda.
Thank you, Benson, for the warm welcome and the introduction. I'm very happy to be here, to be back in at Ari's department to deliver this talk on autophagy. So welcome everyone to this research webinar. Autophagy is actually a very fascinating topic. I find it quite fascinating because it's a cellular mechanism, a cellular process that has been implicated in several health conditions, not only in neurodegeneration, but also in cancer, kidney diseases, and aging. So I hope that after this talk, you will have a clear understanding of autophagy and its role in neurodegenerative disorders and mood disorders. So in this talk, I will start with the description of what autophagy is and its function. I will also discuss the different types of autophagy, focusing on neuronal autophagy, the molecular mechanism of autophagy and its regulation, autophagy in neurodegeneration and mood disorders, also the therapeutic strategies targeting this physiological mechanism, as well as the challenges in therapeutic autophagy. So let's start by understanding what autophagy is and how it works. So autophagy, this word comes from the Greek meaning self-eating. And this mechanism is a highly conserved intracellular machinery in eukaryotic cells. So autophagy actually plays a housekeeping role in the cells, and it's also in charge of getting rid of cellular waste products in the cell and the recycling of cellular components that can be used later on by the cell. So autophagy maintains the cell homeostasis, proteostasis. Proteostasis is just like homeostasis of proteins, and it is also involved in the balance between cell survival and cell death. This is actually a very complex process, and many researchers are trying to understand at the molecular level how the cell can be driven to cell survival or cell death. This is quite relevant in some disorders in which we want to preserve the cell function and integrity to avoid cell loss, like in the case of neurodegenerative disorders. So autophagy is also in charge of the elimination and recycling of cellular components such as damaged organelles and molecules like lipids, nucleic acids, polysaccharides, and proteins. So, and this is quite important to mention because several disorders are associated to misfolded proteins and aggregation of proteins. So for example, in some neurodegenerative diseases, there is accumulation of protein aggregates. And this accumulation of protein aggregates can be due to genetic mutations or impaired proteostasis mechanisms, impaired mechanisms to clear aberrant proteins. And now that we are talking already about protein elimination and degradation, I want to share with you that there are actually two different processes responsible for protein degradation. And the first one is the ubiquitin proteasome system that you can see here. This system consists of a complex, a protease complex that contains proteases for the selective degradation of proteins. So this is a selective process. This is a selective process because the proteins to be degraded are tagged with ubiquitin. Ubiquitin is a small molecule, a small size protein, and this is how the tag proteins are going to get recognized by the proteasome to get degraded. But what happens when the proteins or the organelles to be degraded are too large to go degradation by the proteasome system? Well, then that is the case in which autophagy takes over. This is just to let you know that the autophagy is not the only degradation system in the cell. We also have the ubiquitin proteasome system for protein degradation and for degradation of waste products in the cell. This mechanism of autophagy was first reported back in 1962 in liver cells. And later on, Professor Christian de Duve, he was a Belgian cytologist and biochemist. He coined the term autophagy for the first time in 1963. And actually, he won the Nobel Prize in Physiology or Medicine back in 1974 for the discovery of a very important cellular structure, the lysosome. And he is considered the founding father of autophagy for this discovery. But it was later, until 2016, that the importance of autophagy has been recognized. And this involves another Nobel Prize. This time it was awarded to Professor Yoshinori Yosumi for his research on the mechanisms of autophagy. So he discovered 15 novel autophagy genes. So as a result of these discoveries in autophagy, there has been an increasing number of publications in recent years, as you can see in this graph. And this is because autophagy has been implicated in the pathophysiology of several diseases, such as cancer, liver disease, renal diseases, neurodegenerative disorders, and very recently in psychiatric disorders, such as depression. So to understand the reason for this increasing interest in autophagy, let's take a look at the mechanism of autophagy. So here, this is what happens. The cytosolic substrate or cargo is, this can be damaged organelles or misfolded proteins. This is going to get engulfed in a newly formed membrane. This is called the phagophore, and this membrane comes from the ER or the plasma membrane or even the mitochondria. And this phagophore is going to engulf the cargo through elongation for the degradation of the waste products. And this is known as the elongation phase. So then the phagophore is going to close to form an autophagosome. When we have the autophagosome form, this one is going to travel through the microtubule system to reach the lysosome. So here in green, you can see the lysosome. So there should be fusion of the autophagosome with the lysosome so that the hydrolases containing the lysosome can start the degradation of the waste products. And when there is after fusion of the lysosome with the autophagosome, then this structure is known as autolysosome. So here is when the degradation process takes place. So the cargo gets digested by the lysosomal enzymes, and the products of degradation gets recycled, and they get they are available by the cell again to be used. So for example, the proteins are going to be degraded into amino acids for the synthesis of new proteins. Lipid droplets are going to be degraded to fatty acids, so they can be used to produce energy in the mitochondria by beta oxidation. Glycogen is going to be catalyzed to glucose, and this is going to be used for energy. So the mechanism that I just described is one of the types of autophagy mechanisms, and it's also known as macro autophagy. This is one of the most studied mechanisms. So let's take a look at the other autophagy mechanisms. So in total, there are 3 different mechanisms of autophagy, and the classification of the different types of autophagy depends on the intracellular cargo, how the intracellular cargo is being delivered to the lysosome. Here in orange, you can see the lysosome. So I already described the mechanism of macro autophagy through the formation of the autophagosome that fuses with the lysosome. This is the most characterized autophagy process so far. So when the cargo is sequestered in a non-selective manner, then this process is called in bulk autophagy. But the cargo can also be recruited in a selective manner, and that is known as selective macro autophagy. So within this selective macro autophagy, there are several variants depending on the type of cargo that is being processed. So for example, we have aggregophagy, which is the selective disposal of protein aggregates. Mitophagy, which is the selective degradation of mitochondria. Lipophagy that targets lipid droplets, ribophagy, that is for the ribosome degradation, and chaperone-assisted selective autophagy. So maybe now you might be wondering how the selectivity of the different types of cargo take place. So, well, this selectivity process is not well understood, but what we know now is that there are some molecular players taking care of the selectivity process. So that is, for example, P62 and NBR1. These are recognizing proteins, and both are required for selective autophagy. So what they do is that basically they act as a bridge between the cargo and the autophagy machinery. So for example, P62 is going to bind to ubiquitin in the cargo, and also it's going to bind with components of the autophagy machinery. So that's how this bridging takes place. And NBR1 also interacts with P62. So through this recognition mechanism is how the selectivity of cargo for degradation occurs. The other process of autophagy is microautophagy, and this one is very straightforward because here what happens is that the lysosome is going to directly engulf and take care this take up the cytosolic components for degradation. The other autophagy process is the chaperone-mediated autophagy, and this is also a selective autophagy process. Here, there are some mediators that are involved. For example, the heat shock cognate 70 protein. So this is a chaperone protein. And what it does is that it recognizes a pentapeptide in the cargo. This pentapeptide is also known as a Kieferk motif, and it is special. This pentapeptide is special because of the amino acid sequence and the type of amino acids in this peptide. So what happens here is that the chaperone the chaperone protein is going to bind to the protein to be degraded and translocated to the lysosome through the receptor LAMP2A for degradation. So far, we understand that what autophagy is and the different types of autophagy. So now let's take a look at how autophagy gets activated, like in which situations there is activation of this mechanism, autophagy. So as I previously mentioned, autophagy is an important mechanism to maintain cell homeostasis by the elimination of defective proteins or damaged organelles. So in that way, the cells prevents the accumulation of these toxic components. So autophagy is also activated to meet energy demands. For example, during starvation conditions, autophagy will get activated to recycle cellular components and meet the energy demands. So this is quite important in some other mechanisms, for example, in neurogenesis. And because autophagy keeps this availability of components required for cellular functioning, it also supports neuronal plasticity. Autophagy gets activated during stress conditions, for example, during nutrient starvation, in the absence of growth factors, when there are low oxygen levels. So here, under stress conditions, the main goal is to assist cell survival. Other situations in which autophagy gets activated is during cell development and differentiation. And again, this is quite relevant in some other processes, for example, in neurogenesis. And autophagy can also be activated for programmed cell death. So there are different types of cell death. So the type 1 cell death is apoptosis. Type 2 cell death is autophagy. Type 3 cell death is necrosis. So the excessive activation of autophagy is going to activate the programmed cell death. So autophagy occurs in different types of cells, and now I'm going to focus on neurons. And here the process is called neuronal autophagy. Autophagy plays an important housekeeping role in neurons. Neurons are postmitotic cells. So what happens in mitotic cells is that cell division is one mechanism by which cells can get rid of toxic components in the cell. But this is not the case in neurons because they are postmitotic. So neurons greatly rely on autophagy mechanisms to get rid of toxic products. Autophagy also promotes cell survival and protects against neurodegeneration. So autophagy is considered to have like this neuroprotective effect. And another peculiarity of neurons is that the synapses require high levels of energy and protein turnover. So through autophagy, the neuron can meet the energy demands. Something else that makes neurons special is their morphology, because neurons have large dendritic and axonal cytoplasm. So this kind of makes it difficult to remove damaged organelles and protein aggregates in time, because it takes time for the autophagosome to reach the cell body. In the cell body, the lysosomes are found. So the autophagosomes have to travel a long distance along the axon to reach the lysosome so that the degradation process can start. So here you can clearly see how autophagy is very important in neurons and why neurons are sensitive to the disruptions in this process. So neurons are susceptible to cell damage and neuronal loss due to dysregulated autophagy. So now I would like to spend some time to dig into the molecular mechanism of autophagy in neurodegenerative disorders. Yes, so to understand how the abnormalities in autophagy can lead to neurodegeneration, we need to have a look at the regulation mechanism of autophagy. And here I want to warn you, because maybe the next slide is a little bit shocking because you will see a lot of regulators, but don't worry, I will break it down and explain each step in detail. So autophagy, as you can see here in this figure, is a highly regulated multi-step process. It involves induction, elongation, the autophagosome formation, autophagosome trafficking, and autophagosome and lysosome fusion and degradation. So here let's start with the induction phase. In the induction phase, a key initiator of induction or player in the induction phase of autophagy is the mammalian target of rapamycin or mTOR kinase. So mTOR is a kinase, and it's a master regulator of cell metabolism and cell growth and cell proliferation, and it also it plays a role in the induction of autophagy. So this protein is actually part of a nutrient sensor system, which is called the mTOR complex 1 system. And this nutrient sensor system controls protein synthesis, and it is regulated by insulin, growth factors, amino acids, by mechanical stimuli, and oxidative stress. So what happens here is that mTOR activation triggers anabolic processes and such as protein or lipid synthesis, as well as the stimulation of energy metabolism. And autophagy is actually the opposite. Autophagy is a catabolic process. So what happens here is that mTOR activation inhibits autophagy. So in other words, mTOR, in other words, mTOR activation indicates the cell to grow and proliferate, and at the same time, it inhibits autophagy, while mTOR inhibition is going to promote autophagy. The next, also another player here in the induction of autophagy is a kinase, the AMPK, which activates autophagy mechanism by the phosphorylation of this other protein, ULK1. And AMPK is a protein kinase that is activated in response to stress. So for example, when there is low glucose or low oxygen levels, and also during heat shock. So the activation of autophagy can be done either by inhibition of the mTOR system or by activation of the AMPK. So this is going to phosphorylate this protein, ULK1, and this protein is part of a complex, as you can see here. And the activation of this protein is going to activate another protein, the AMBRA protein, that is part of the PI3C complex. So this is how induction of autophagy takes place. So the next phase is the elongation phase. So let's take a look at what happens here. In this case, the phosphatidyl inositol 3-phosphate, PI3P, it's generated from the activation of the PI3K complex. And this PI3P is going to bind to other proteins. Let's call them WIPs. The name is too long. It's a WD repeat domain phosphatidyl inositol interacting 1 and 2 proteins. So what the WIPs do, they catalyze reactions that regulate membrane elongation through autophagy proteins, as we can see here. So after elongation, then the next phase is the autophagosome formation. And here there are some autophagy proteins involved. So the autophagy protein 5, 12, and 16, they form a complex. And this complex is going to assist the conjugation of phosphatidyl ethanol amine to the LC3 protein. And this facilitates the closure of the autophagosome. So when the autophagosome is formed, the next phase is autophagosome trafficking. And actually this step is very important in neurons. And in this step is it's when the autophagosome gets transported to reach the lysosome. So it's very important in neurons because the autophagosome has to travel long distances along the axon to reach the lysosome in the cell body. So the machinery for autophagy trafficking includes cytoskeletal system, such as actin and the microtubules, the motor proteins, such as myosins, and other proteins like LC3, which is present in the membrane of the autophagosome. And all these proteins help the autophagosome move along the microtubule system so that it can reach the lysosome. So here, actually, the regulation of autophagy trafficking is not well understood, but this step, as you can see, is very important in neurons because of the distance that the autophagosome has to travel for cargo degradation. The next step is the autophagosome lysosome fusion. So here in this process, the membrane tethering factors play a role. They promote membrane fusion, and they do it by creating bridges to bring together the two membranes. So the membrane of the autophagosome and the membrane of the lysosome. And there are several players involved here. For example, the Hoops complex, ATG14, and the RAPGTPases, as indicated here in the figure. Another important component in the fusion step is the snares. These are proteins that are present on the membrane of the lysosome and the autophagosome, and they form a 4-helix bundle as shown here. So when this, another player here actually is also the ATG 14 protein, because it assists with the interaction between the snare components. So when the snare components are found on one membrane, it is called the cis snare complex. And when they are found in two different membranes, like for example in the autophagosome and also in the lysosome, it is called the trans snare complex. And maybe this structure looks a little bit similar, because what happens here is that the snare components are going to act as a zipper, bridging together the two membranes. The hoop complex, on the other hand, is formed by the association of other protein subunits, and this hoop complex interacts with proteins from both membranes, as you can see here, to form a stable complex and promote the fusion of the membranes. Also, the hoop complex is important for pore formation, because the components of the lysosome need to go and reach the cargo in the autophagosome. So the pore formation is quite important. So far we know that autophagy is a multi-step process, that each step is highly regulated, that there are multiple players involved, that in some steps the regulation process is not well understood. We also can see that disruption of autophagy can occur at any stage of the autophagy process, and some of these steps are actually affected in neurodegenerative disorders, as we will see in the following slides. So let's start with Parkinson's disease. In most cases of Parkinson's disease, the cause is unknown, but in 5% of the cases, and there have been genetic mutations identified. So these mutations affect autophagy at different stages. And one of those mutations have been found in the protein alpha-synuclein. So alpha-synuclein is a presynaptic protein that regulates vesicle trafficking and neurotransmitter release. So in Alzheimer's disease, there is abnormal accumulation of this protein, and it aggregates in dopaminergic neurons in the substantia nigra, what we call this aggregation is known as the Lewis bodies, and this leads to cell loss. So in this picture, you can see an immunostaining of the alpha-synuclein in the Lewis body. Okay, so what happens here is that in healthy conditions, alpha-synuclein is going to be degraded by chaperone-mediated autophagy mechanism. So it enters into this degradation system. But the mutant alpha-synuclein is going to bind to the LAMP2A receptor and it blocks it. So that's a problem here because this blockage is going to affect the degradation of alpha-synuclein and the degradation of other products that also undergo this degradation system. So when this happens, there is an upregulation of autophagy by the accumulation of alpha-synuclein. So this excessive autophagy activation and long-term autophagy upregulation is going to activate the type 2 cell death. So that will lead to cell loss. Also, the accumulation of alpha-synuclein is going to have an effect on another type of autophagy mechanism. We already saw that it has an effect on the chaperone-mediated autophagy mechanism, but it also affects macro-autophagy. So what happens here is that the accumulation of alpha-synuclein affects the elongation stage in macro-autophagy because it mislocalizes the autophagy protein 9, and this affects the autophagosome formation. Mitophagy dysfunction has also been associated to Parkinson's disease, and in this case, it involves two proteins, Parkin and PINK1. So what happens is that in normal conditions, Parkin participates in the translocation of damaged mitochondria for clearance. So it does that by binding, by labeling the mitochondria with ubiquitin units. So this leads to the recruitment of other autophagy components. And PINK1, what it does is that it accumulates in the outer membrane of the damaged mitochondria to recruit Parkin. So this occurs in the initial phase of mitophagy to clear damaged mitochondria. But some mutations in Parkin and PINK1 have also been identified in Parkinson's disease, and the mutant Parkin has been found in early onset of Parkinson's disease is disrupting the clearance of damaged mitochondria. Also, PINK1 affects the recruitment. The mutant PINK1 is going to affect the recruitment of Parkin, leading to impaired mitophagy. Another process that is also involved in or a contributor to the loss of dopaminergic neurons in Parkinson's disease is oxidative stress. So by decreasing oxidative stress through autophagy, this has been considered a therapeutic strategy. Also, some antioxidants such as compounds that contain thiol groups, ascorbic acid, and polyphenols can induce autophagy and reduce oxidative stress. So some antioxidants have been shown neuroprotective effect to autophagy activation. And now that we are talking about some therapeutic options, let's take a look at other compounds that also have been tested in vitro, in vivo, and in clinical trials to assess their autophagy enhancer properties in Parkinson's disease. So for example, resveratrol. This is a compound found in food, in grapes and in berries and peanuts. This compound induces autophagy by the activation of AMPK and the inhibition of the mTOR system. Also, there's another compound, nilotinib. This is a tyrosine kinase inhibitor. And this compound increases the clearance of alpha-synuclein by the upregulation of Beclin-1 and the autophagy protein ATG12. It also increases the interaction of Parkin to promote mitophagy. Another compound is Trehalose, this is actually a naturally occurring disaccharide, and it increases the clearance of alpha-synuclein by the upregulation of ATG10, Becklin-1, and ATG12, and it also upregulates lysosomal hydrolases. Another example here of an autophagy enhancer in Parkinson's disease is a compound piperine, and this compound is extracted from a Chinese medicine with anti-inflammatory properties and antioxidant effects, that is the piper longum. And what piperine does is that it inhibits the mTOR system. So now let's take a look at another neurodegenerative disorders and how autophagy is involved in the pathophysiology of Alzheimer's disease. Alzheimer's disease is characterized by the presence of two abnormal structures, that is the senile plaques that are formed by beta amyloid peptides and the tau containing neurofibrillary tangles. In this picture, you can see an immunostaining showing the senile plaques and the neurofibrillary tangle. So the beta amyloid peptides get generated by the enzymatic cleavage of the amyloid precursor protein, as you can see here in this video, which is a transmembrane protein. So it gets cleaved by secretases and resulting in the beta amyloid peptide. And this beta amyloid peptide accumulates, forming the plaques. So the amyloid precursor protein is actually involved in synaptogenesis and synaptic plasticity. The extracellular domain of the amyloid precursor protein also acts as a growth factor, and it promotes neurogenesis. But the proteolytic process of the amyloid precursor protein releases the beta amyloid peptide, which is neurotoxic, and it is centrally implicated in Alzheimer's disease. As I previously mentioned, tau is another protein that has been implicated in Alzheimer's disease. So tau is a microtubule-associated protein that promotes microtubule self-assembly. And its function is to stabilize the microtubule, and it also controls vesicle transport in neurons. But the abnormality in tau occurs when it gets hyperphosphorylated by tau kinases. So then it generates the phosphorylated tau, hyperphosphorylated tau, which is prone to form aggregates. And these aggregates form filaments, and then that is how the neurofibrillary tangles are formed. And these are characteristics of Alzheimer's disease. Okay, so there is actually growing evidence that autophagy might be involved in the pathogenesis of Alzheimer's disease because of the accumulation of autophagy vacuoles that has been observed in Alzheimer brains. And these autophagy vacuoles contain the amyloid precursor protein and the products of the enzymatic cleavage of this protein, including the beta amyloid peptides. So this is an indication of impaired autophagy. Another piece of evidence pointing out at impaired autophagy is related to the trafficking and lysosome function. So what happens here is that the transport of the autophagy vacuole along the axons is affected, it's interrupted. So that leads to accumulation of the autophagosome, and the autophagosome accumulation of the autophagosomes actually enhances beta amyloid production. So beta amyloid peptide seems to regulate autophagy by inducing the formation of autophagy buckles to the stimulation of the AMPK and the activation through basic Becklin 1. So the beta amyloid peptide enhances autophagy. And this actually makes sense because this peptide is neurotoxic. So that is a kind of signal that is being detected by autophagy so that this machinery can get rid of the beta amyloid peptides. But it's not possible to do that because the autophagy machinery is being affected. Also, lysosomal function is impaired in Alzheimer's disease. And in this case, there is a protein involved, presenilin 1, which is a catalytic subunit of alpha secretase. Remember that alpha secretase is involved in the cleavage of the amyloid precursor protein. So there's a mutant presenilin 1 that affects directly lysosomal function. But now I already explained what happens with the beta amyloid peptide and in autophagy. So let's take a look at what happens with tau. Tau is found in different forms. For example, truncated tau as a monomer, as an oligomer, and also the phosphorylated form of tau. And most of these forms are cleared by autophagy. So in Alzheimer's disease, what happens is that there is hyperphosphorylated tau, but the clearance of hyperphosphorylated tau gets impaired. So that's why tau gets aggregates and it forms the tau-containing tangles that are found in Alzheimer's disease brains. The truncated tau is also cleared by autophagy, and the accumulation of truncated tau has been observed in Alzheimer's disease, which indicates failure in the autophagy-mediated clearance of tau. So now let's take a look at autophagy enhancers that has been tested in Alzheimer's disease. And one of them is rapamycin, which is an antibiotic, and it is a very well-known inhibitor of the mTOR pathway. So it activates autophagy by inhibiting the mTOR system. And it has been observed that it ameliorates the beta amyloid and tau pathology in animal models of Alzheimer's disease. Another autophagy enhancer in Alzheimer's disease is latrepirtin, which is an antihistamine drug, and it stimulates autophagy, it promotes autophagy by stimulating ATG5, by upregulating ATG5. There is another compound that is also being tested, and that is a protein phosphatase 2A. This induces autophagy in Alzheimer's disease. And this enzyme, what it does is that it removes phosphate groups. And remember that the phosphorylated tau is involved in the pathology of Alzheimer's disease. So removing the phosphate groups in the phosphorylated tau promotes its clearance. Also, lithium has been observed that it induces autophagy by stimulation of MK. Another compound is nicotinamide, which is a form of vitamin 3, and this compound induces it reduces autophagosome accumulation by upregulation of LC3, and it's also involved at in the function of the lysosome. So that is how the autophagy process gets promoted, or stimulated. There are other neurodegenerative disorders in which autophagy has shown to play a role. I'm not going to go into detail, but very briefly, in amyotrophic lateral sclerosis, this is a motor neuron disease that leads to respiratory failure. It has been observed that there is a mutant superoxide dismutase 1 that is cleared by autophagy. And in Huntington disease, this is also another motor neuron disease. The mutant Huntington protein aggregates, and this protein can be removed by autophagy. So as you can see here, autophagy plays a crucial role in neurodegenerative disorders, and in recent years, it has also been linked to depression. So let's see what happens in depression. Depression is a multifactorial disorder in which the different factors contribute. For example, environmental factors, psychosocial factors, and genetic factors. So when we talk about the molecular mechanism of depression, we need to keep in mind that the pathophysiology of depression involves complex signaling networks. So that's why different hypotheses have been developed to understand depression. For instance, the monoamine deficiency hypothesis, the neuroplasticity hypothesis involving neurogenesis, the stress hormone system, among other hypotheses. And the most recent hypothesis that has emerged to understand the pathophysiology of depression is the autophagy hypothesis. So there has been very recent evidence associating autophagy and depression, but the molecular correlation of autophagy and depression is still at its infancy. It's not very well understood. So in neurological disorders, it's actually very clear that autophagy contributes to the degradation of neurotoxic molecules, and that has a positive effect. But in depression, what has been observed is changes in autophagy, but how those changes correlate to depression is still under investigation. So also the molecular mechanism of autophagy in depression is still not well understood. But what we know now is that autophagy is changing in when there is treatment with antidepressant. Actually, the first indication of autophagy involving depression comes from the treatment with antidepressants. So upon treatment with antidepressant, there is induction of autophagy, and that has been observed. So for example, a fluoxetine treatment phosphorylates ULK1, and by now you already know that ULK1 activation results in induction of autophagy. Also, another piece of evidence that autophagy is implicated in depression is that inhibition of Beclin-1, remember this in the induction of autophagy mechanism, induces depression-like behavior. Also, exercise has been observed that it enhances autophagy and it induces depressive symptoms. There is also evidence that autophagy increases neurogenesis, which contributes to antidepressant effect. So some autophagy modulators in depression include, of course, different types of antidepressants, for example, tricyclic antidepressants, serotonin selective reuptake inhibitors, fast-acting antidepressants, and other drugs, such as rapamycin, iperazine, which is a major compound found in St. John's wort, and St. John's wort is used as an antidepressant. This is a herbal medicine. Also, silibinin. This compound is extracted from another plant, Silybum marianum, also known as milk thistle, and electroconvulsive therapy also triggers autophagy in the hippocampus. But as I previously mentioned, the knowledge of the involvement of autophagy in depression is still under investigation. So there are so many questions at the moment, For example, at what level autophagy is impaired in depression, at what level antidepressants act on autophagy, what is the crosstalk that is taking place between neurogenesis and autophagy, what is the regulatory mechanism of autophagy during the different stages of neurogenesis, or for example, what is the autophagosome and the autolysosome content at the different stages of neurogenesis. So there are so many questions, and these are actually challenges that we are facing right now in autophagy research in neurodegenerative disorders and also in depression. For example, another thing that we need to keep in mind is that increased expression of autophagy markers and activation of autophagy mediators do not translate into autophagy function. And that is something that we need to keep in mind to interpret the results, you know, because here we need to consider autophagy flux, like that is the measure of autophagy function. So in that way, we can know that, okay, autophagy is being induced, but actually the whole process of autophagy, which is a multi-step process, is not interrupted. It's functional. Autophagy is functional. So another thing to consider here, and another challenge, is that autophagy stimulation in neuropsychiatric disorders might be a neuroprotective strategy, but we need to keep in mind that we need to avoid excessive stimulation of autophagy, because if autophagy gets overstimulated, then we can trigger cell death mechanism. So it's something to keep in mind. And there's also a lot to do in terms of research regarding treatment dose and treatment duration of autophagy inducers. So that has to be examined as well. So with this information, I would like to conclude my talk.